Hello, in this video we will discuss about the MAP kinase signaling pathway and RAS, RAF and ARC and MAC pathway we will discuss also. So briefly overview we will discuss about the MAP kinase. So what is the MAP kinase signaling pathway? So basically the MAP kinase signaling pathway is the family of pathways. Basically is the family of pathways which that is lead to cell proliferation and programmed cell death, also the programmed cell death. Remember about that. Cell proliferation and programmed cell death. If it means the cell if damaged, so the programmed cell death will be occur. If that will be not occur, so it means the lead to cell proliferation and cause cancer. And so the cancer progression and development. It is used for the development also and growth. So let's begin to understand without wasting any time. To understand, here is I draw the plasma membrane, which that is basically inside the plasma membrane is the cytosol, while outside is the interstitial part. So here is you can see that is their plasma membrane and cytoplasm, and here is the basically tyrosine receptor tyrosine kinase is the monomers. So two monomers is contain a receptor tyrosine kinase. It's contain a cytoplasmic domain and receptor domain. Receptor domain are present in the outside of the cytoplasm, and the cytoplasmic domain are present in the inside of the cytoplasm. So basically is the family of pathway which that is lead to control the different mechanism to cell survival or cell growth or cell death. So here is the basically the family of pathway which that we will discuss also. The four type of that pathway we will discuss about the cell proliferation as well as the stress and apoptosis. So that means the basically increase or decrease the cell progression. So here is the cascade of the phosphorylation due to the kinases. So this kinase is used for the phosphorylation. So the phosphorylation of cytoplasmic protein is here, which that is the cascade of the phosphorylation of the kinases to lead a transcriptional factor. So I hope you make sense about the pathway, which that is basically horizontally shows that the members associated events, while the vertically shows that the cascade of activation events. So here is the member associative events contain a horizontally line which contain a same kinases, which that is the same type of kinases, while the vertical, if we will talk about the one line of the cascade of activation, we can get a different type of kinases. So I hope you make sense about that. Horizontally basically is the similar, but the changes can be. So the horizontally we can classify by the stepwise. So that's why phosphorylation will lead to cascade of the another protein triggering to activation of the pathway. So here is the members associated events. Here you can see that, and that is the map kinase or map 3 kinase, which that is the basically is the same type, but that is the basically is used for the different events. For the horizontally is the cascade. So here is the map kinase is the 7 members, which that is the third option. You can see horizontally. So that is the map kinase is the 13 members are present. That's map kinase. So the map kinase is the 13 members. So here is the first is the phosphorylation of cytoplasmic protein to give a ARC1-2 pathway, which that is the transcriptional factor for cell proliferation and growth and differentiation. But on the other hand, the map kinase 13 members are present, which that is basically Here is the transcriptional factor ARC1 to 2 pathway, which that is used for the cell proliferation and growth and differentiation. It means if the damaged cell will lead to ARC1 to 2 pathway to cause mutation and cause cancer. Why? Because it is progression phase and proliferation of the cell division. So on the other hand, here is the GJNK pathway, which that is stress and apoptosis lead to stress and apoptosis, while the P38 pathway is the same stress and apoptosis. And here is the ARC5 pathway used for the cell survival and anti-apoptosis differentiation. So it means when the ARC5 pathway will be enhanced, it means the if tumor suppressor gene will not activate to inhibit this mechanism, so our production of ARC5 pathway will lead to cause the cell proliferation to cause cancer. So here is the nucleus, which contain a nuclear pore and the nuclear membrane, which that is also the bilayer of the phospholipids, hydrophobic outside, while the inside is the outside the hydrophilic, while inside the hydrophobic. So here is contain a chromatin material, genome, which that contain a chromosome, and this chromosome is made up of histone protein and histone protein 8 histone protein, which contain a 4 type. Wrapping around the DNA is known as the nucleosome. So here is the DNA, which contain a gene. This gene is the specific for the specific pathway. So for example, the P38 pathway, which that is used for the stress and apoptosis, that's lead to produce the after the transcriptional factor bind with the specific gene site of the tumor suppressor gene. After the tumor suppressor gene binding to increase and transcription of the messenger RNA from this gene of the DNA. After this messenger RNA, this pre-messenger RNA modified by the exon and intron separation to move outside in the nuclear pore to produce the protein P53 and P21. These P53 and P21 will lead to inhibit the cell growth, so that stress and apoptosis will be occur. While on the other hand, let's begin to understand basically when the here is the receptor domain, which that receive the signaling molecule. After the binding of the signaling molecule, the receptor tyrosine kinase will be bind with the monomers. So these monomers will be bind. After the binding to cytoplasmic domain, that is attract to each other, and dimerization, and so this was the RAS RAF MEC ARC signaling pathway initiation. So that's after the dimerization, dimers form. Dimers, mere means the part and di mean two. So the tyrosine kinase domain will be remain in the cytoplasmic domain. So that is the basically cytoplasmic domain bind. After this bind, here is the another step is known as the trans-autophosphorylation. This trans-autophosphorylation is autophosphorylation. So the autophosphorylation means the phosphate group will be attached to changes, the conformational changes in cytoplasmic domain. After this conformational change in cytoplasmic domain will lead to phosphorylation of the GRB2 protein. This GRB protein basically is bound by the phosphorylation of the kinase enzyme. This kinase enzyme will transfer the phosphate group to the GRB to activate. This is the basically phosphorylation due to the attachment of the phosphate group of the cytoplasmic domain. So the cytoplasmic domain will give the phosphate group. So remember about that. After this, basically the phosphate group will be give the activation of the GRB2. So the next one is the source. Source is the basically the protein which that is inactive in the form of GTP attachment. So the kinase enzyme, another kinase enzyme used to source is basically the son of 7 less guanine nucleotide exchange factor, which that is basically the son of 7 less guanine. So this is the sunless guanine nucleotide exchange. Basically, the ATP will give this to GDP convert into the GTP. So that will be source will be activated and bind with the GERB2. After this binding, you can see and you can watch in this next step to understand better. So GERB2 will be bind with the phosphate group and source will be also bind with the GERB due to the activation by the guanosine triphosphate. After the binding, so the GDP, basically the GDP containing RAS protein are present in the peripheral region of the inside of the cytoplasm, so that will be activated by the GTP, which that is attached with the source will give the phosphate group to the GDP to convert the GTP of the RAS protein. So the RAS protein is basically the small monomeric G protein, which that is used for the cell proliferation and cell growth. So that will be inhibit the cell apoptosis. While in the vice versa, we can say that the tumor suppressor gene will be inhibit this mechanism. We will discuss later in this video. So here is the small monomeric G protein. It is also known as the RAS protein. After the activation by the GTP attached from the source, which that gives the phosphate group to the RAS protein to convert the GTP RAS protein, it means the phosphorylation. This phosphorylation is due to the kinase enzyme. So the basically phosphorylation. So after this phosphorylation, it means the another phosphorylation of the RAF, MAP kinase basically is also phosphorylation, and this phosphorylation is basically the phosphorylation to attach with the GTP contain RAS protein. That is the basically is the mechanism. Here is the MAP2K kinase is the type of this, which that is here is the MAP2K, MAP kinase. That is the basically horizontally is the members associated events. So here is the members associated events is the MAP kinase MAP2K is the 7 members. So anyhow, here is the RAF will be bind, and next is the MAP kinase is the basically is the MAC pathway. This MAC pathway is phosphorylation. Cascade of the phosphorylation will be bind with the RAF protein. After this binding, that's complex formation will be occur. So here you can see in this figure that is the RAF will be a RAF pathway is contain a MAP kinase mechanism, which there is a phosphorylation, and this phosphorylation basically the kinase MAP3 kinase basically is the, and that's next one is the MAC that is the MAC pathway is the MAP kinase associated protein, which that is lead to the phosphorylation and bind with the RAF pathway, which that is the MAP kinase, which that is after the binding. The next one is the MAP kinase pathway, which that is lead to basically the ARC pathway is lead to MAP kinase protein. This MAP kinase protein will lead to bind, and after this binding, that's close to each other, and the maturation of the ARC protein, this phosphorylase. So this ARC protein is become a transcriptional factor, which that is transcribe the gene to translate the protein for the requirement. For example, the cytoplasmic target, phosphorylated cytoplasmic target, and enzyme kinase, and cytoskeleton part, and regulator of apoptosis. So this is used for the apoptosis part. It is regulate. Basically, it's inhibit the apoptosis. So let's begin to understand here That will be the gene expression by the transcription factor to nuclear target to cell cycle and cell growth and cell proliferation and inhibit the apoptosis. While on the other hand, that was the GNK pathway and uh, P38 pathway, which that is used for the stress and apoptosis. But this is not. This is inhibit the apoptosis. So let's begin to understand in summary. Here is the RAS, which that is activated by the GTP from the GDP, and that's a RAF basically is activated by the phosphorylation and phosphorylation by the protein MAP kinase and the MEC pathway, which that is activated by the MAP kinase protein phosphorylation and the ARC pathway, which that is basically the MAP kinase enzyme protein, which that is basically is give the pathway. So that's all pathways the medically is inhibited by the medicine, which that is basically ARC pathway lead to cause cancer. So it 17% cancer is due to the RAS activation. So this RAS activation, but why? Because the tumor suppressor gene is methylated or mutated due to the methylation or some DNA damage. So that is why the tumor suppressor gene is not working and not inhibit the RAS protein. It means the overproduction of the cyclin and CDK cyclin dependent kinase. So here is the Fanib, which that is the medicine used for the inhibition of the RAF, which that is the different type, and TNIP is the used for the MEC inhibition. So the different type of the different, for example, the binimatinib and silumatinib, and on the other hand the dabrafanib, which that is used for the RAF inhibition. So this was the pathway which that is the kinase is used for the transfer of the phosphate group to another protein, that is the cascade mechanism. So here is the tumor suppressor gene. This tumor suppressor gene is also present in the nucleus basically. So after the transcription of that, due to the give the signal due to the overproduction of the RAS protein, RAS activation will lead to produce by the transcription to produce that protein P53 and P21, which that is lead to inhibit the RAS activation. So that will be inhibit. So it means the cell apoptosis will be occur. So that cell apoptosis will be occur due to the inhibition of the RAS. So the activation of the P38 pathway, stress and apoptosis, and JNK pathway, which that is also lead to stress and apoptosis. So the 70% mutation for cancer due to RAS gene activation. So remember about that. On the other hand, the tumor suppressor gene is also lead to inhibit the activation of the tyrosine kinase receptor domain, which that is inhibited by the signal attachment. That signal is basically is important for the cell growth, but important thing is that when the DNA will be damaged, so it will no need to cell proliferation. So if cell will be proliferate due to the ARC pathway, so that means it will lead to cause cancer. So thanks for watching. That was the overview of the pathway.